Is it all right if I stand down here? Perfect. Okay. My name is Lauren Goodrich. I'm excited to kickstart this conference today. I come from Roche. It's a site in Madison that was not originally a peptide site. And I'll talk about that in just a little bit. But what I want to talk about today is our peptide microarray technology and the ability for us to screen libraries of millions of linear and cyclic peptides using these microarrays. So as I just mentioned, our Roche site is in Madison, Wisconsin. It started as a startup out of the University of Wisconsin, Madison. And the core technology was a DNA microarray technology. It was commercialized by the startup NimbleGen Systems in 1999, and 7 years later it was acquired by Roche. And at that point they sold DNA microarrays. Technically, because of our DNA history, we're a member of Roche Diagnostics. We do collaborations with Roche Pharma, but technically we are also open to discussion with external parties. And I'm just going to keep flying because we only have 15 minutes and I'm sure I'm going to get cut off. So, our technology is a light-directed maskless microarray synthesis technology. So it's a fully automated process that combines UV light, digital micromirror devices, which are shown here on the left, and photolabile protecting groups on the N-terminus of amino acids. So basically, we shine UV light on this digital micromirror device. So these are the same DMDs that are found in your 1080p TVs, probably this overhead projector, although I'm not sure where it is. But it's essentially just a grid of mirrors. And so depending on the version of the DMD, so for a 1080p DMD, it's actually a 10 micron by 10 micron mirror. And depending on the angle of this mirror, it either directs light onto the microarray surface or into a dead space, essentially. And so using these digital micromirror devices, we can spatially control photolabile protecting groups on a microscope slide. So here's just a quick schematic of our synthesis. Here, the little purple balls are our photoprotecting groups on the N-terminus of each growing peptide chain. So in this particular example, we're turning light on in the second and fourth mirror position, which deprotects the N-terminus of the peptide. We couple the next amino acid, which here I believe is asparagine, although it's a little fuzzy. And we essentially at this point have regenerated our photolabile surface. So then we deprotect light. We deprotect again in the next synthesis cycle. So here maybe we want to deprotect the first and fourth position, couple the next monomer, and repeat this process until we've generated a complete complex library of peptides. So we're using these peptide arrays for several applications, both for diagnostics and pharma. I'm not going to talk at all about the diagnostic applications, but we're doing a lot of work, and we have a peptide service lab in the area of biomarker discovery and antibody characterization. But since this is a therapeutics conference, I'm going to spend time today talking about our drug lead discovery and optimization. And I think the message I really want to drive home today is that this is more than a screening platform. So yes, we can initially screen a library of peptides against drug targets, but then we can also use this technology to do the optimization of those lead peptides directly on this platform. So again, discovery and optimization of peptide molecules on these microarray surfaces. And here are some of the really brief reasons we think it's really good for this purpose. One is the high density of the mirrors on this DMD device. So on one fully automated microarray synthesizer, we can generate 18 million peptides on a single microscope slide, there's my pointer, in a 24 to 48 hour run. That's on one synthesizer. We have multiple synthesizers running. So this isn't by any means the upper limit. It's just an example of one running synthesizer. We can generate very diverse chemical libraries. So we generate both linear and cyclic peptides. And because it's a fully chemical synthesis platform, doesn't rely on any biological systems, we can incorporate all kinds of non-natural amino acids. So of course we use the 20 standard L amino acids, but then we also incorporate D, alpha, beta, gamma, other backbone modifiers. And this is just a really brief, kind of crude schematic of our chemical library, but we have over 300 monomers in our library that are photoprotected on the end term of each amino acid. And you can see from this pie diagram, the natural amino acids are really a small fraction of this. And so the diversity of chemical libraries we can generate is quite massive. In addition, we synthesize peptides with N-alkylation, so N-methylation and peptoids. And then the last point I want to make on this slide is that because we have the spatial address of every peptide on this microarray slide, our platform is really sensitive. So we don't only gain information about the top hits in a screen, we also get information about some of the lower level hits that we can maybe optimize to have higher binding affinity. So we like to really stress the sensitivity of this platform to detect weak binding interactions that we can drive to be higher affinity binders to these protein targets. And I'll talk about that a little bit. So here's just our general workflow. A scientist typically sits down, designs our library. We pass this off to a bioinformatics scientist who essentially generates a list of rules for our synthesizer. The synthesizer runs in a completely automated way to synthesize a microarray. And this happens, it really depends on the design. Some are complete in a matter of like 8 hours. Others take a little longer, so 48 hours. At this point, a labeled drug target is incubated on the microarray surface, and this label is fluorescence. So we scan the microarray for fluorescence, and this is an actual microarray image shown here. We align the image and extract fluorescent intensity information, analyze the results for potential families that we may see emerging, and then at this point, we can take hits and rapidly optimize them. So we put it right back into the workflow where we design another library based on the hits we found the first time. And then we synthesize a microarray, reproduce, and repeat this process. Okay, so today I want to share a really brief story that we just published. It's kind of old work, but we finally got around to putting it together in a paper. And it's a process we called Stepwise evolution. And it's a way that we essentially screen for linear peptides where we look for a hotspot core of interactions between a peptide and a protein target, and then grow this peptide to increase affinity. So we really start with low affinity interactions and drive these to high affinity interactions. So I'm going to tell you that story. And then I want to really briefly talk about how we're able to incorporate things that would make these better drugs. So things like cyclization, non-natural amino acids, backbone modifiers, et cetera, such that we can make libraries of drug-like molecules. Okay, so here's our process of what we call stepwise evolution. We start with a 5-mer, so we make all possible 5-mers using the 20 standard amino acids. Our rationale for this is twofold. One, it fits perfectly on a single microarray with with 2 replicates. And the second is that the chemical space gets really big really quickly. So you can't imagine synthesizing a 15-mer with every possible amino acid combination effectively. And so we start with a small peptide and grow this peptide. So we search for hotspots, which are our 5-mers. Then we extend in 2 amino acids on the N and C term. And then we go through and substitute and mature this peptide. This essentially takes 3 peptide microarrays. And if you remember our workflow, this can happen quite rapidly. And so using only 3 microarrays, we can find really potent lead compounds. Okay, so the target we used for this example is Streptavidin. Of course, it's not really of therapeutic interest to most people, but it's a protein that a lot of people have used to test screening compounds or screening technologies. And the important thing I want to point out is that the most frequently and basically only reported hotspot for this target is an HPQ or HPM motif. And so the question we asked when we decided to design this experiment was, can we find other motifs that also bind to Streptavidin, and or can we mature these such that they have higher affinity than the standard HPQ motif? Okay, so here's the data from a 5-mer array. So we've synthesized all possible 5-mers on a microarray. And what you're looking at is fluorescent intensity with 1 replicate on the y-axis and a second on the x-axis. If we look at the nature of these peptides, you see all the highest affinity binders. And so this is the type of thing you pull down from another screening technology, be it mRNA display, phage display. But you see all the top hits are this HPQ or HPM motif. If we bioinformatically remove all of those top hits, what we start to see emerge are other families of peptides. So we see this LH motif, a PAWAH, and this FDDWL motif also show up. And yes, they're lower affinity than the HPQ motif now, but can we make them better affinity is the real question. And so we go through the extension process where we put 2 amino acids on the N term and 2 amino acids on the C term. And what you're seeing is essentially a ranked list of all of the binders that come off of that microarray. At this point, we go through a substitution or maturation step. And this plot looks really confusing, but I'm going to take a second to walk through it. It's basically a souped-up alanine scan, where instead of just substituting each position of the peptide that we've discovered to alanine, we substitute it to all other 20 amino acids, where the sample key is over here. So for example, in this fourth position, where this is the HBQ motif, there is nothing else you can substitute this to gain binding affinity, which is shown on the y-axis here. So the only way you get signal intensity in this particular motif is by maintaining the histidine. That's not true for other positions like this threonine or glutamine. So what you really get from these types of scans is where the peptide is interacting with the protein target. We've shown that these also correlate really well to crystal structure data, which I don't have time to talk about today since we only have 15 minutes, but I'm happy to discuss offline. So we've then crystallized all of these peptides with streptavidin just to show that they indeed do bind to streptavidin. The known HBQ motif is a simple loop design, and they are all overlaid on one slide, so it's a little messy. But you can see the other three families of peptides we found, so in the green, orange, and red. And what's really cool is that we found a really diverse chemical space in terms of scaffold peptides. So even with a peptide as short as a 5-mer, we're able to start selecting for chemical structure, which is pretty cool. The other thing I want to point out here is that we were actually able to take one of our low affinity binders and drive this to nanomolar affinity for streptavidin. This is the first time we're aware that anyone's found a nanomolar binder to streptavidin. We've also done this with D amino acids. So this is a complete D peptide shown here, where we again took hotspots, extended and matured. Affinities are shown here as well. We've also done this with a cyclic peptide. So this is a head-to-tail cyclic peptide, mixture of L and D amino acids. The affinity here is, of course, not good, but this was the first hit out of a screen. And I'll show you here on the next slide where we're able to, by just incorporating a few other non-natural amino acids, drive the affinity of this molecule. So this NQD-proline W, which is a crystal structure head-to-tail cyclic I showed you a couple slides back, through the incorporation of a few non-natural amino acids, we're able to find other families of peptides that bind to streptavidin in the cyclic form with much higher affinity. And we haven't crystallized any of these just because this is just our test case. So basically, I hope I've convinced you that we can generate cyclic peptides with non-natural amino acids. I've shown you a few backbone modifiers as well in terms of the D amino acids. But we're really driving these libraries now with therapeutic targets, which unfortunately I can't share much of that with you today. So a couple other things we're working on that are of interest. We're designing sets of permeable libraries with Professor Scott Loki. And basically what we're doing is we're taking known permeable scaffolds that he's discovered in his lab at UC Santa Cruz and varying side chain chemistry to find binders to therapeutic targets. So this is really interesting, and with our vast chemical library, I think we have a real chance at finding some permeable peptides in the near future. We've also done a lot of work in terms of enzymology, determining substrates for particular peptides. One particular example is site-specific conjugation, published recently. You can look here. But basically, the enzyme is transglutaminase. It's known as a meat glue in the food industry. And this is because it cross-links glutamine and lysine to form an isopeptide bond. And so the thought was that we could use this for site-specific conjugation if we could determine the substrates for each of these residues, both glutamine and lysine. And we did just that. So you can look into that in the paper. So I might have actually made it in 15 minutes, which I'm shocked. We flew. Okay. So again, peptide microarray platform. This is a completely automated process. We have robust QC protocols, high reproducibility. What I really want to drive home again here is the speed with which we can screen and optimize these peptides. Complete chemical synthesis, so linear cyclic peptides, L, D amino acids, other non-natural amino acids. And with that, I'm happy to take questions.
Let's thank Laura.